So Simon studied at Cambridge and then was at Addenbrooke's and qualified, moved to Nottingham for his basic surgical training and came to Oxford in 2006, taking up a position with the evidence base in transplantation, and completed his thesis, and then was appointed as honorary consultant transplant surgeon and senior clinical research fellow here in October 2017. So, and I'll introduce James at the same time. James is an academic clinical lecturer in transplantation and has a contract with University Hospital Coventry and the University of Oxford, looking at live donor kidney transplant and complex vascular access. And this is the first appointment to the Coventry-Oxford Transplant Network, which is a UK transplant collaborative, which is aimed at improving patient access to transplant by sharing resources to the centres. So I think we're starting with Simon. Thank you very much.
Right, morning everybody. Thank you for coming. I wrote the title before I'd actually thought about what we were going to talk about this morning. So actually we're going to focus on kidney preservation, having talked quite a lot in the past about the liver. And so I'm going to talk about some of the clinical research that we've been doing and part of, and then James is going to talk a little bit more about some of the translational and lab research. So for those of you that work at the Churchill, this is a fairly familiar site outside the theatres, particularly if Sanjay's been on call for the weekend. That's a post-Sanjay weekend on the right-hand side there, which is the mainstay of how we store kidneys in the UK at the moment. So we put them in ice boxes, so cold fluid in ice, cooled down to about 4 degrees. And this is what we've been doing probably for about 30, 40, 50 years in the UK. Since we started transplanting routinely in the 1960s, 1970s. And there's a very good reason for it. It's very simple, it's very cheap. Logistically, it's straightforward, it doesn't require power, there are no moving parts to break, and nobody has to sit with it and watch and make sure that it's doing what it should do. But it does have its limitations. So when you store an organ at 4 degrees, metabolism is slowed, but it's not stopped completely. And so there's still anaerobic metabolism carrying on in the organ, you get accumulation of metabolites, you get depletion of ATP, and so when you put the organ in and you reperfuse it in your recipient, you get an ischemia-reperfusion injury, you get inflammation, which then primes the immune system with a risk of inflammation and damage. There are lots of reasons why we might want to reconsider this idea of cold storage. So over the last 10 years or so, as you can see from this graph from NHSBT, our donors have been gradually getting older. Older donors generally have more pre-existing injury, so from hypertension, diabetes, cardiovascular disease. And so we're starting off with slightly more marginal kidneys than we would have been perhaps 10, 20 years ago. Equally, the type of donors that we're using has changed as well during that period of time. So our ideal donor, our ideal deceased donor is a brainstem dead donor because there's no period of warm ischemia prior to procurement of the organs. We're seeing now and using now more and more donors after circulatory death, DCD donors, where there's a mandatory period of 5 minutes of warm ischemia before you can start the retrieval operation. So these organs are more injured before we even start this preservation process. So before we start looking at some of the tools we've got available to us, it's worth thinking about what the ideal organ preservation method looks like. So obviously we want to prevent damage during storage, but we also ideally want to be able to repair some of the damage that's already occurred during that procurement process. We'd like to be able to prolong the storage time for logistical reasons, so we can transport organs longer distances or we can accept more organs at any one time. But also we'd like to be able to assess the viability of organs. If we could have some way of trying to work out which organs are going to behave, work when we transplant them, that would help us to select better and possibly use some of the organs that we don't currently use. And then moving on into the future, we'd also like to be able to deliver therapeutic interventions to the organs before we transplant them. So cold storage doesn't stack up too well against all of these criteria. Probably does help to slow organ damage during storage, although it doesn't stop it completely. But it doesn't really allow any of the others. It doesn't repair any damage, storage times are limited, and there's no means of viability assessment. So even back when we started this in the 1960s, there was an interest in machine preservation of organs. So this is Belzer's hypothermic machine preservation device. So this essentially pumps cold fluid through a kidney, and as you can see, it's pretty unwieldy and logistically challenging. It's required a transit van to take it around, a forklift truck to move it, and so perhaps unsurprisingly, as cold storage solutions improved during the 60s and 70s, people moved to cold storage for the logistical benefits. Nowadays, the machines have changed somewhat. We can now put it on a tabletop and one person can lift it, and this is the equivalent that we have now in our theatres. And again, this is a hypothermic machine for perfusing kidneys with cold preservation fluid. Now, the benefits of this, or the reported benefits of this, is you're flushing cold preservation fluid through the kidney during storage, so you're removing some of those metabolites that are accumulating and providing substrate at the same time. So when he was with us, John O'Callaghan undertook a systematic review looking at hypothermic machine preservation, and there clearly is some clinical benefit to using the machine over standard static cold storage. So these are all randomized controlled trials of hypothermic machine preservation versus static cold storage. As you can see, the risk of delayed graft function is reduced by about 20% when you use hypothermic machine preservation. But interestingly, there wasn't much difference in graft survival or graft function when you use this technology. So it does seem to prevent the reperfusion injury, but it doesn't have too much of an impact on long-term outcomes. Most of you have heard Rutger talk before about the COPE Consortium. So this was an EC-funded consortium of transplant centres across Europe who were looking at organ preservation research. And as part of that, there were 3 randomized controlled clinical trials and a suite of experimental research as well. David, at the last grand round, presented the results of the liver trial, the normothermic liver preservation trial. The next of these trials to report is the third one down there, the COMPARE randomized controlled trial, which was looking at hypothermic kidney preservation. And the results of this have just come out. So the idea behind this trial is to see whether we can improve on hypothermic machine preservation. So typically we flush the kidneys with cold preservation fluid, but there's no oxygenation. We're just literally flushing metabolites out of the kidney. The group in Poitiers, who were part of the COPE Consortium, had done some experimental work, which is shown here. Essentially, they took a DCD pig model, so 60 minutes of warm ischemia to the kidney, removed the kidney, placed it on a hypothermic machine preservation device for 22 hours, and then transplanted back into the pigs again. And they split the organs into 2 groups. One group was standard hypothermic machine preservation, the type that we're used to using in clinical practice. The second one, they actively oxygenated the perfusate. So there's no oxygen carrier, but they're just bubbling oxygen through the perfusate that they're using in the device. And you can see that there's a much lower peak in creatinine after reperfusion in the transplanted animals, and the baseline creatinine falls down much more rapidly to a lower level. And then if you take biopsies 3 months after transplantation, there's much less in the way of chronic inflammation and fibrosis in the organs. So oxygenation does seem to have some benefit in preventing both short-term and long-term damage. And so the purpose of the COMPARE study was to test this in a clinical setting. So this was a multi-center randomized controlled trial across Europe looking at oxygenated hypothermic machine preservation versus standard hypothermic machine preservation. One of the nice things about the kidney is that they come in pairs. So each donor has 2. So it's quite a nice clinical trial design to actually just take both kidneys from one donor and treat one way, one another way, and then you can control for all of those donor factors. So this was a paired kidney design study. It was blinded. So the way this worked is we had a small army of medical students and other students in the Netherlands and Belgium with vans who would take these devices out to the retrieval hospital, switch the oxygen on or off depending on the randomization code, put the kidney on the device so that they could adequately blind the surgeons in both the retrieving and implanting centers as to whether there was oxygenation or not. And this study was in these more marginal kidneys, so DCD donors aged over 50. The primary endpoint was graft function. Okay, so 1 year after transplant, we looked at estimated GFR. In the primary analysis, you can see there's a slight drop in GFR in the standard hypothermic machine preservation group, and it's improved in the oxygenated group. It doesn't quite reach statistical significance. The problem with using graft function as an outcome is, of course, if a kidney is not functioning 1 year after transplant, you can't include it in your analysis. And so as a pre-planned sensitivity analysis, we imputed all those grafts that have failed with a GFR of 10 mils per minute, and that's shown on the right-hand side, and that shows that if you take into account the graft failures, the difference does reach statistical significance. If you look at measured GFR, so we also measured GFRs in these patients at 12 months, again, a statistically significant and probably clinically significant difference of 7 mils per minute in GFR at 1 year. Perhaps more interesting is a significant reduction in graft failure in the oxygenated group. And if you look at the mechanism for that, you see there's a halving in the risk of acute rejection during the first year post-transplant with oxygenated machine preservation. So there does seem to be something in this. The clinical data does support that animal data from Poitiers, that 1 year after transplantation with oxygenated machine preservation, we're seeing improved function, less graft failure, and a reduction in the risk of acute rejection. And it seems to be that reduction in the risk of acute rejection that seems to mediate the differences in function and graft failure. In terms of the mechanisms, well, if you're oxygenating, you will reduce the risk of ischemic damage, build up of metabolites during preservation, and so you get less of an ischemia-reperfusion injury. And if you reduce the ischemia-reperfusion injury, there'll be less inflammation post-reperfusion, which will result in less priming of the innate immune response and the subsequent adaptive immune response. And we seem to be seeing less in the way of acute rejection, but also less in the way of chronic inflammation and fibrosis later on. So hypothermic machine preservation then, if we add oxygen to it, does seem to do a little better than cold storage. We can prevent damage during storage, we can repair damage to a degree that's occurring during preservation, which might allow us to store kidneys for a little bit longer. But we still don't have a means of assessing viability of organs during preservation to see which ones we think are going to work or not. In order to be able to do that, we've really got to be able to store our organs in a functioning state, and that's where normothermic preservation comes in. So we're now perfusing the kidneys with oxygenated blood at body temperature, providing nutrients so that they can metabolize as normal, which has the potential to allow us to assess viability and deliver therapeutics on the circuit. David Nasrallah in our last ground round presented the results of the liver study using normothermic machine preservation, and certainly in the liver there does appear to be a reduction in the reperfusion injury following transplantation, but perhaps more interestingly there was also a difference in discard rate between the two arms in that study, which suggests that the surgeons were able to use information from the device to make a decision as to whether they could transplant a liver or not, and potentially improve utilization of livers that would otherwise be discarded. So what about the kidney? Well, you can't really talk about normothermic preservation of the kidney without talking about the work of Mike Nicholson and Sarah Hosgood, who are now in Cambridge, who've really done most of the initial clinical research in this area. So they have a slightly less developed circuit that you can see on the right-hand side there, consisting of a bunch of pumps and an oxygenator and derived from a pediatric anesthetic machine, I think. And they took 18 kidneys that they'd accepted otherwise for transplant, and they placed them on this circuit for an hour just after cold storage prior to transplantation, and then transplanted the kidneys. And you can see that they get quite impressive delayed graft function rates when they do this. So we would expect in kidneys like this to see a delayed graft function rate of perhaps 30% to 50%. And they saw in their 18 kidneys a delayed graft function rate of just over 5%. So this does seem to be having some effect in repairing the organs prior to reperfusion. What about viability assessment? So they took this one step further, and they took a whole bunch of discarded kidneys, so kidneys that were not accepted for transplant anywhere in the UK. They took these kidneys, they perfused them, and they developed what they call the QAS score, which is a sort of clinical score for assessing organ viability based upon macroscopic perfusion, blood flow, urine output. They then applied this score prospectively to a further group of discarded kidneys, and demonstrated that they were able to transplant some of these kidneys that were declined by all UK centres and actually improve the transplant rate. And so the 5 kidneys that they transplanted, 4 had immediate function, and they all did okay after a year. So the challenge with their method is that they're only perfusing for a very short period of time. So they're only perfusing for an hour immediately prior to transplantation. So you're not preventing any of the damage that occurs during storage, but they do seem to be able to reverse some of that damage prior to transplantation. This clearly doesn't have any logistical benefits because you're not prolonging preservation time, but it does seem to allow viability assessment. So off the back of this and the liver research, Anne-Marie Weisenbacher, when she was here as a DPhil student, along with Peter and Konstantin, developed a prototype normothermic kidney preservation device based upon the liver circuit that we've used previously. And Anne-Marie used firstly a series of pig kidneys and then discarded human kidneys, and was able to demonstrate that she could perfuse them under stable conditions for up to 24 hours, so far longer than anybody else has managed to this point. So this shows the development of the device. There's Konstantin and Anne-Marie on the right-hand side there. So it started off as a bunch of lab equipment and turned into a quite nice-looking prototype, which has been developed further since then. One of the challenges with the kidney is that, unlike the liver, the kidney produces urine. And so if you leave it on a circuit for too long, then it will obviously produce urine, the circuit volume will decrease, and so somehow you have to replace that volume. So some of the early experiments that Anne-Marie did, she added in Ringer's lactate to try and maintain the circulating volume, but you find that very quickly during preservation that you get electrolyte imbalances, pH imbalances, and it's very difficult to maintain perfusion for long periods of time. And so one of the things that she tested during her experiments was this idea of urine recirculation. So you simply take the urine that the kidneys excreted and add it back into the circuit to maintain homeostasis. And that actually seems to work quite well. So the graphs here, the black dots are the kidneys without urine recirculation, and then the red ones are the ones with urine recirculation. And you can see that you can preserve a kidney without recirculation just using Ringer's lactate for perhaps up to 6 hours or so, but then you start to get electrolyte and pH imbalances, and eventually the resistance increases, the perfusion pressures decrease, and you're unable to maintain perfusion for longer periods of time. With urine recirculation, you can get a nice stable preservation for 24 hours or even longer. So normothermic perfusion seems to tick most of our boxes. We can prevent damage, we can repair damage, we can potentially store for long periods of time, up to and beyond 24 hours, and we potentially have means of viability assessment and enough time on the circuit to be able to deliver therapeutic interventions. So where are we taking this next? Well, we've got funding from the NIHR I4I for a phase 1 clinical trial of this prototype device. And the aims of that are to assess safety and feasibility of NMP for the first time in human kidneys for up to 24 hours. At the same time, we'll collect samples to look for biomarkers for perfusion and predicting viability of donor kidneys, and to look at the post-transplant outcomes of these kidneys. We'll compare them to historical controls. So we're going to take 36 kidneys. We're going to perfuse them in gradually increasing durations from 6 to 12 to 24 hours. And there'll be a safety stop after each phase of the trial to look at the results and make sure that, that things are working as we expect. And then each patient will be matched to historical controls from our own cohort to see whether there's any impact on clinical outcomes. Primary outcomes, purely a safety outcome, 30-day graft survival, and then we'll also look at graft function, longer-term graft survival, and the incidence of delayed and primary non-function. And then we'll also be taking samples looking at perfusion parameters and biomarkers as potential predictors of organ viability. So I'm going to hand over to James now, who's going to talk about some of the more sort of lab and translational research.
Hi, everyone. Thanks very much, Simon. So I'm going to talk a bit about the science side of what we've been doing in the department. And it seems like this might be a little bit back to front, but they kind of run in parallel. And I'm going to go back over a little bit of background as well, just so it fits in with the science as well as the clinical. And so, I mean, as transplant surgeons, our objective is to transplant more organs. We don't want to compromise patients' outcome. And we want better graft survival at a year. We want fewer kidneys that don't work. And I've shown a graph on the left side, which, if you ignore the green line at the bottom, shows the outcomes over 5 years of all deceased donor transplants. And actually, if you look at the 1 year numbers, so if you look here, you've got around about 90-95% survival at 1 year. So the survival rates are very good. They're not good enough, we want them better, but they're very good. And in fact, they're so good that drug companies have almost stopped doing clinical trials into immunosuppression because you can't really power a trial to go from 95 to 96, 97% without many, many, many patients. So we need to try and find better ways of improving these outcomes. And so the way we've been doing this is Simon showed this graph, and I apologize for the repetition, but the question is there a need to do this? Well, as he explained, the donors are getting older, and there's not much we can do about that. And if you look, about 280 patients last year died whilst waiting for either a kidney or a pancreas transplant. So there certainly is a need to do this. And where can we find the resource for this? We're transplanting what we think is as many kidneys as we're offered, but actually we're not. So this is consistent over 10 years. About 10 or 12% of kidneys that are retrieved are not transplanted. So of those, we think about half of them are probably transplantable. So how do we go about assessing this? And when we get this, what we think the problem here is, what's in the box? Okay, well, that's in the box. It's a kidney. And for those non-transplant surgeons, it looks okay. You see that kidney, you think, I'll transplant that, that's great. It looks fine, it's well flushed, someone's very kindly benched it for us. But what we don't know, once it's transplanted, is it going to be this or is it going to be this? Because you can't tell from just looking at the kidney like that. And so that's where the normothermic perfusion ideally comes into play. And Simon talked about Mike Nicholson's work, which has got some, which is fine if you just take some very simple measures, but there's nothing else in there that tells us about what's inside the kidney. And so our aims in the department were both clinical and scientific. The clinical, the funding for the phase 1 trial has been achieved. Thereafter, a funding for a larger randomized trial to see where this technology can be used. And then a conditioning unit. That's the goal. We have kidneys that come from around the country or from wherever they've not been transplanted, and we assess them. And then we decide whether they're transplantable, and if they are, we either transplant them ourselves or we send them elsewhere to be transplanted. And in parallel with that, we've got some scientific goals. So using discarded human kidneys for research is challenging because they're a very heterogeneous group. There's a huge variation in what you get, and Anne-Marie's data were fantastic, but some of these kidneys had gone around the houses and had very, very long periods of cold ischemia. And so we needed a better model to assess the science than the discarded human kidneys, because we wanted to look at things like mechanisms of action, and we wanted to add novel therapies to see where that might take us. And so if you think Mike's machine looks a bit Heath Robinson, then you can just take a look at ours. And I've put this up because this was very much in the early days where we've got a sophisticated pump and the technology that you can't see there that actually does the work. But this is a device where you pump from a centrifugal pump blood into the oxygenator, up into the renal artery here, and then it drains down the renal vein and then back around the circuit. And we've modified that because if you just buy those disposables, they're €2,000 each. So if you imagine we go to our abattoir, we get a pig kidney, we get some pig blood, which is essentially free, and then we need €2,000 to do the experiment. So that's completely unrealistic in any walk of research. And so by doing it this way, we can reuse components, we can buy them separately, we can do it in a way that we can actually get some information by pumping large numbers of kidneys without spending lots of money. And the closed circuit is the one that we started with, and then this is what's called the open circuit, because the vein is not cannulated. So the renal vein is draining directly into the chamber full of blood, rather than having it suspended in a, as you saw on the previous. And so one of our early questions was, well, which one's better? Does it matter what we use? Because the open circuit is much, much easier to use than the closed circuit. It's much less of a faff. And so we had a medical student who came and did some experiments with us, and we just compared the two. This was right at the bottom of our learning curve 3-4 years ago when we first started. And so it's not perfect experiments. But what it sort of showed initially was there wasn't much difference in terms of perfusion characteristics on the machine. The closed circuit seemed to produce more urine. Now, there's a theory that if you've got a cannula in the vein, it's a bit like having it attached to the IVC. You've got some venous pressure. Whereas if you've got the renal vein draining into a canister, there's no venous pressure at all. So maybe you increase the pressure across the glomerulus and you help that. That's possible. But actually, when we looked at the kind of markers of injury and the other parameters, it wasn't consistent. So the lactate was higher in the closed circuit, the LDH was higher in the open, and so in the end, for the sake of ease, we went to the open circuit. Okay, but with these pig kidneys, we could not perfuse them beyond about 8 hours. The human kidneys seem to work okay. Pig kidneys are much more sensitive, they're much more vasospastic. And so we couldn't control the CO2 levels. And in parallel with what we were doing, there was a Canadian group who just did this beautiful pig auto-transplant model where the kidneys were pumped for 16 hours, transplanted, and the results were fantastic. So we thought, goodness me, what are they doing that we're not? Why can they do it and we can't? And so we just made some small changes. We changed things like the vasodilator, okay? And so this is 3 groups. We used to use sodium nitroprusside because that's what Mike Nicholson used, that's what others had used, and it seemed to work. Our colleagues in Groningen, I'm glad Rutger's not here because he would tell me off for not pronouncing it right, Groningen in the Netherlands, they don't use any vasodilator. And the CATS group, they use Verapamil. So we compared the 3. And what we found is there wasn't much difference, interestingly, even if you didn't use a vasodilator at all. The sodium nitroprusside kidneys look like they produce more urine, but if you look at the error bars, it's essentially one kidney will just be an outlier there and produce lots of urine. But what we found was striking, because the sodium nitroprusside kidneys, which is the vasodilator we've been using all along, got a profound lactic acidosis, and the pH drops dramatically, okay? And so you do a little bit more digging, a bit more research, and you find that one of the metabolic byproducts of sodium nitroprusside is a cyanide-type compound, which interferes with oxidative respiration and effectively stops the kidneys respiring. So we stopped using that, and we started using Verapamil. And then we had a fantastic Italian nephrologist who came over and spent a year with us. So this is the Italian influence, and we wanted to tinker a bit with the preservation solution we were using, because as you heard, Simon said, the Nicholson group used Ringer's lactate, Anne-Marie used albumin, human albumin, and in the liver trial, gelafusin was used. And so there's so many questions that need answering. Which is the best one? Which is the right one to use? And so what we did here is we took the pig blood that came from the abattoir with the kidney, and we split it into its component fractions, and then we resuspended the red cells with a known amount of plasma. Took out all the noise, took out all the immune cells. And we're a little bit more meticulous about some of the sterility, and if we filtered the blood and there were still white cells in it, we filtered it again. And what we found is the perfusion got better. And so suddenly we got abattoir pig kidneys, and we could perfuse them for 18 hours, which is what we've been wanting to achieve as a sort of model of what we wanted to compare with the human setting. They produce urine, the perfusion was reasonably stable, they are acknowledged at the end if you look, the lactate does start increasing, but that's 1 or 2 kidneys. And so, if you don't believe me, I thought I'd show you some nice pictures. So this is a kidney on the machine. It looks shiny because it's covered in cling film, which again is a very cost-effective way of keeping the kidney warm and moist. And so at 1 hour, it's reasonably pink and perfused, and at 18 hours, the same. So you can pretty much tell when a kidney's dying because it starts looking black and blotchy and horrible, and that kidney looks okay. So we had started to achieve this objective of a research model of pig kidneys that was a cost-effective way of doing it. And so we wanted to look a bit more into some of the mechanisms of action. We know that mitochondria are a crucial part of the cell. We know there's hundreds of factors that are important in all of this, clotting cascade, etc. But we wanted to look at the mitochondrial function in kidneys undergoing normal thermofusion. And so this is funded by an Academy of Medical Sciences grant, and we used animal house pigs, not slaughterhouse pigs for this model. Okay, and so pigs were anaesthetized, had a laparotomy, and then they had one kidney removed and flushed, completely healthy. It's a beautiful living donor kidney. This is the gold standard kidney. It's an uninjured kidney, and it's just flushed and put on ice temporarily. The contralateral kidney is then clamped for 60 minutes to give it a 60-minute warm ischemic hit. So it's pretty injured. You can see how injured it is. You compare that picture to the one before where the kidney looked nice and pink and healthy. So this is a pretty ischemic kidney. And then on top of that, we wanted to put it on ice or on the pump for 24 hours. So it has a period of warm ischemic injury where it's got deoxygenated blood in it, and then it's got a period of cold ischemic injury where it's sitting in a box of ice. And then we perfuse both of them, for 8 hours on our machine. Okay? So what we found initially, well, if you compare the two groups, interestingly, there doesn't appear to be much difference. So the Nicholson score tells you flow is important. And we looked at this and thought, but it doesn't seem to matter. But of this injured group, 2 kidneys didn't make it to 8 hours. They made it to 4 or 5 hours, and they were essentially dying. It was a terminal model. The kidneys looked purple, poorly perfused. And so the graph doesn't really give the game away at all. So we split the groups, and we split them into injured kidneys that appeared to recover, healthy kidneys, which is the blue line, and then the grey line at the bottom is those kidneys that did not perfuse and did not survive and were not viable. And the lines start to split. So perhaps flow is important. But more crucially, when we start looking at measures that aren't just perfusion parameters like oxygen consumption, you might think it's logical and obvious, but actually we haven't seen this before. The oxygen consumption drops dramatically. They stop consuming oxygen in these kidneys that start dying, and their lactate levels go up. So we start to find objective measures of viability during NMP other than just blood flow. And the other interesting thing is, despite the urine output of these injured kidneys, they're not functioning. Okay, so they're not clearing creatinine. If you look at the orange line, no creatinine clearance at all. The healthy kidneys, as you would expect, pretty consistent creatinine clearance. Same with the urine albumin creatinine ratio. So what that shows is leaky glomeruli due to injury just leak albumin into the urine. So the healthy kidneys don't do that, and the injured ones do. So measuring function is not very helpful because they're not functioning. It doesn't tell you whether they're viable or not. It just tells you that they're not working. It doesn't tell you whether they'll recover. So we looked at the mitochondria. And so the healthy kidneys, the graph on the left, the mitochondrial respiration is pretty stable. And the way you do this, just out of interest, if anyone is particularly interested, is you take the kidney biopsies at different time points, you homogenize the tissue, and then you extract the mitochondria fresh. So you have to do this at the time of the experiment. You can't just sort of put it in the freezer and say, maybe we'll do this next month. So it's quite time and resource intensive. And once you've got those mitochondria extracted, you put them in a chamber, Clark's electrode, and you pass oxygen into it. And then you measure the current across the chamber. And it tells you how much oxygen they are using at that time. And so it's a measure of mitochondrial respiration, a measure of mitochondrial function. So it's probably the most accurate way of assessing how these mitochondria function. And so the healthy ones just sort of keep functioning. The injured ones, although it's only small numbers, there is a suggestion in those mitochondria that manage to get to 8 hours that they start recovering. So the graphs, you'll have to believe me with your eye of faith, starts going up. But we acknowledge this is small numbers, and we acknowledge it's a suggestion rather than any significance. So the other thing you notice is that the levels are a bit lower. So if you look at the y-axis, we've got just below 20 here, and we've got around about 50 here, which sort of fits with an injured kidney. And then we split the graphs again to see whether the injured kidneys that didn't perfuse well were different to those that did. And apart from perhaps this anomalous reading here, the injured kidneys that don't make it, sort of the mitochondria appear not to function as well as the ones that are either healthy or the ischemic ones that recover. So we had some suggestion that mitochondrial function is important during this, which again, it makes sense, doesn't it? You need mitochondria, but this was work that hadn't been done before. And so then I'll just touch briefly on some of the science that Anne-Marie did. So this is one of the human kidneys that was discarded on the left. And you can see, the picture doesn't come up very well, but it's quite patchy here. And this kidney would have been turned down for perfusion characteristics. A transplant surgeon somewhere would have looked at it and said, no, that's not well perfused, we're not transplanting it. Actually, when you put it on the machine, it looks okay. Sorry about the microphone. And so we wanted to look at whether you can demonstrate any regeneration or repair during perfusion. Hepatocyte growth factor is a marker that's produced, it's a protein produced by many different cells, not just the liver, which is involved in tissue regeneration, tissue repair. And so across perfusion, you can see the levels increase. So the kidney appears to be producing levels of hepatocyte growth factor. It appears to be able to produce proteins, which demonstrates its functioning to some degree. And this is a clever scatter plot. This is work that others did. It's on kidney tissue from Anne-Marie's experiments. And it essentially shows the change in protein expression across the perfusion. So the left-hand one is a scatter plot, and it shows the difference in upregulated proteins and downregulated proteins throughout the perfusion. Each dot's a different protein. The heat map on the left is just a different way of demonstrating this. So it goes from 0, 14, 24 hours, and each line is a protein, and red doesn't mean good and green, red doesn't mean bad and green means good. It just tells you whether it's been upregulated or downregulated. And again, that doesn't mean it's good or bad. It might be a heat shock protein, it might be a marker of inflammation, it may be a repair protein. It just tells you that there's a change. And so what we see during perfusion is we see a change in the protein profile of the kidney, which again, that's fascinating to know. This helps you with determining pathway analysis, and it helps you with hypothesis generation. You look at these proteins and think, where do we look next? How can we find out what's going on? So this is crucial work. And then we decided to have a bit of fun and add some therapies you've got a kidney on a machine, you can do what you like. You can add whatever you like to it. It's a fantastic model for testing things on. So I'm not going to steal Caitlin Rosenberg's thunder, because she did a lot of this work for her DPhil and hasn't presented that work yet. But we've got a collaboration with called the MEEP Collaboration, which is between a group in Aarhus in Denmark and Rotterdam in the Netherlands, where we produce pig-derived or adipose-derived pig stem cells. And we've been doing the normothermic perfusion side of that. So the initial bit was a dose testing. So you take 2 million, 10 million, and 50 million stem cells, and you give them to a kidney during machine perfusion. And the questions were very simple. Where do they go? Can we find them? These questions are relatively unanswered. Do they have any effect? If so, what effect? So we weren't trying to be too clever. We just wanted to see if we could answer some simple questions. As part of this, there's a pig auto-transplant model. The results are currently being analysed. I'm not going to talk about that today, but that's a part of the sort of bigger picture with this project. And so these graphs just show the groups of control, 2 million, 10 million, and 50 million stem cells. And so essentially what it showed was it didn't really matter how many stem cells you give them. It doesn't seem to change what happens during perfusion. So the perfusion characteristics were the same. The injury markers were the same. The hemolysis, the lactate, they were all essentially the same. There wasn't any difference in anything that you measured. Okay. The perfusion tended to dip a bit when you gave lots and lots of stem cells, but it seemed to recover. And so is that positive or negative? Well, it means you can give lots of stem cells. It doesn't appear to clog up the capillary system. It doesn't appear to have an effect on perfusion. So that may be positive because it means you can give lots of stem cells if they work. or it also means you don't have to give lots of stem cells. But we don't really know what they do. And so we have done some work looking at whether it improves cytokine release and growth factors. I haven't got time to go into all of that, but because I want to show you some nice pretty pictures, because that's more interesting. So we took some biopsies from these kidneys to see if we could find the stem cells. And these are labeled, these stem cells, with something called Q dots. And the slightly perverse side of this is, what you do is you take this big population of stem cells which have been expanded in a very clever way, and you add Q dots to them, and then that affects their viability. And they probably don't work. But if you don't add Q dots, you can't find them. You've got no idea where they go. So for this experiment, we added these because we wanted to see where they go. So it's probably not fair assessing how well they work and trying to find where they go in the same experiment. But this is a slide of a kidney, and this was the 2 million stem cells group. And you can see one. Can you see one? Good. So we found one, which was, that was fantastic. We found a stem cell. Good. Well done us. Now, one Q dot doesn't mean one stem cell, because some of the stem cells take up lots and lots and lots of Q dots. And then if the stem cell dies, it releases the Q dots. So actually, it tells you that there's probably a stem cell there, but there's definitely a Q dot there, and a stem cell has been there at some point. But then if you go on and look at the 10 million, there's more. Some here, and down there. And they're in the glomerulus, interestingly. That's where they all appear to be. And then you look at the 50 million, and there's more still. So it's not quantified, but there appears to be a dose response. So the more stem cells you give, the more you can find. And again, that's interesting. That's useful. This has not been done before. Again, they appear to be in the glomeruli. That's probably because they get stuck in the capillaries. We don't know for sure, but they go into the glomerular blood supply, and then they probably get stuck in the endothelium because they're bigger than the caliber of the capillary. Makes sense. And so this work will lead on to more. There's lots more facets to this, but this is just to give you an idea of some of the things we're doing and where we're taking the preservation work. And so overall, in our kind of progression towards both these clinical and scientific goals, Simon's told you that the COPES trials have been completed, the hypothermic oxygenated perfusion. The funding for the phase 1 trial has been complete. And we've started to work on and produce a reproducible model of kidney perfusion on using abattoir pig kidneys, which is cost-effective and can be used, reused consumables. We've got some promising data on mitochondrial recovery. We've got lots to do on MSC therapy, and we've got plenty of work ongoing. And we've just got a little grant to look at perfusion solution. We've got some work on MRI imaging and giving dyes to the kidney. So there's lots and lots of work to be done here. So thank you very much for listening. I hope that's been interesting. We've got lots of collaborators, lots of people to thank. I hope I haven't missed anybody. If I have, I'm sure they're not here. Happy to take any questions.